Hello my wonderful PU2 champions out there. I hope all my little friends out there are doing great. And I also understand that this season is pretty torturous because there are so many exams ongoing. But my dear children, have that self-belief. I believe in you and you guys are absolutely rock stars. You will be able to make it up. Yes? Now, about this video. So we all saw the thumbnail. It says that in biology, 8 chapters are very much enough for scoring 60 plus marks. Is that true, ma'am? Can we actually get 60 plus marks with just 8 chapters? The answer is yes. The answer is definitely yes, and I hope you are ready for the magic. But before that, I need to tell you one very important thing. That is, you should always focus on 2 things. One is hard work. Definitely you should do it. But along with that, please understand that there is something known as smart work. And it is also equally important. So hard works moves you forward, but smart works is the one that will be helping you decide in which direction. Now suppose the direction is being lost, obviously you will be ending up stranded. So please understand that if at all you are someone who just sits and grinds without understanding what are the things that actually I should be learning first, then you are doing something really wrong. Your hard work is being put at a wrong place. So please don't do that, and I am here to help you with that. So here are those 8 magical chapters, my dear children. This is put down. This is according to the blueprint of 2025-26. As you can see, you have sexual reproduction, flowering plants, human reproduction, reproductive health, principles of inheritance, molecular base of inheritance, evolution, human health and disease, microbes in human welfare. Now reproductive health is a chapter which is easier. I consider it's an easy chapter. So out of 8 chapters out there, 1 chapter is super easy. There are few other chapters also which you have been studying from your first exam onwards. So from the first unit test that you gave, then you also gave midterm for the same chapters. I am pretty sure that at least if at all you are not very 100% confident about those chapters, you are somewhere in the 50%, 60% range where you think that yes, I know it a little bit, but definitely I have to study those chapters. So I have taken only those chapters over here, and I'll tell you how these 8 chapters, if at all studied properly, can give you 60 plus marks. So if at all you have seen our question paper, my dear children, from the 5 marker category, we have to answer 5 questions. So there are 2 sections of part D. Okay, there are 2 sections out there, and at both of these places you have to attempt 5 marker questions. So in total, if we do, there are 5 marker questions that we have to attempt. Now, if at all I see these chapters, okay, principles of inheritance and molecular base of inheritance gives 2 5 marker. Again, here also it is 2 5 marker. Suppose these 2 chapters I have studied it really nice, then I can attempt both of these questions from both of these chapters, right? So 4 5 marker questions are easily done. And now suppose from any of these chapters, suppose human health and diseases or human reproduction, you are able to attempt 1 more 5 marker. What will happen? You have attempted the 5 marker questions out there. Now if you think that my molecular base of inheritance is not that strong, okay, but I'll still work on it, okay, in that case you can borrow some question from here and here, that is from human health and diseases and sexual reproduction in flowering plants. Anyway, you have options being given over here. That means if at all you study these chapters, you would have enough options to choose as well. Now if I think I will only study these 2 chapters, there are chances that you may not be able to attempt these questions out there. These 1 of 2 questions might be tough for you. In that case, what will you do? So please have some extra options, which is why I have taken all these chapters. Otherwise, I could have given 6 chapters, 5 chapters, this chapter, that chapter, but it does not work like that. So assuming that I have solved 5 marker questions, so basically it means I have got 25 marks already. Now from the 3 marker category, again I can just solve 5 questions. Let me randomly take any 5. So human reproduction chapter, again I took 1 question from here, 1 question from reproductive health because it is easy, very easy question. And now human health and diseases also I take 1 question. So 3 questions are being done. Now 4th question I take from microbes in human welfare, and I have evolution as well as sexual reproduction. Suppose I take 1 question from here, my 5 questions are being done. I have one more option out there. So in that case also 3 into 5. 3 into 5 is equals to what? You have got 15 marks. So please tally it. 25 plus 15 is already how much marks? It is already 40 marks out there. Now going ahead, in the 2 marker category also you have an option to write 5 answers. Now over here, you have 1, 2, 3, 4, 5, 6 option. Out of that you can choose 5. So suppose I studied this chapter, since I am solving a lot of questions from this chapter. Sexual reproduction is very easy for me now. Reproductive health was already easy for me. So 1, 2 done. Okay, now microbes in human welfare, I have taken 1 from here, and now I have these options. So anyway, I have to cover these chapters. 1, 2, 3, 4, 5. Again I have an answer. Again I have an option for all of these things. Then what do you say? 2 into 5 is equals to 10. So we were at 40. 40 plus 10 is equals to 50. Now 1 marker, how many ever 1 markers are there? There are almost 15 1 marker questions out there. Okay, after that you have fill in the blanks 1 marker question. So 20 in total. So 15 MCQs and 5 fill in the blanks question, in total 20 marks. So let me see how can I solve it. So over here, I just have to calculate 1 plus 2 is equals to 1 what? So 1 plus 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So this gives me 13 marks. This gives me 13 marks. So 13 plus what did you have? 50. 13 plus 50 is equals to what? 63 marks I am easily getting from these chapters. Now ma'am, so you are saying I should study only these chapters and go? No. I am saying if at all you are a person who is not very much confident with what you should be doing as of now, okay, or you are having that starting trouble, please go ahead and complete these chapters very, very nicely. Very, very nicely, such that you are 100% confident that any question that comes up from this chapter, I am going to solve it. And my dear children, we are doing Brahmastra series. It is a super important series. I do not know how to tell you the importance of it. It's really, really important. The previous year when we did it, so many children got the benefit of it. I do not know if this year children are taking it seriously or not. If at all you are someone who had been watching Brahmastra series, kudos to you. You are absolutely rocking, because this you are at the right place, okay? And if you are not, it's the high time to start watching it as well. It goes for all the subjects. I can specifically guarantee for my subject, because I make my PPT, I know what questions I included as well. Now if at all you want a bonus question, my dear children, bonus chapter, there is biodiversity and conservation as well, where if at all you attempt 2 more questions, so it will be 65 also. Okay, so it's a very easy chapter, which is why I have put it over here. There's no other reason to put it. Okay, now we'll go ahead and talk the important topics of all these chapters that you should not be missing. But before that, if at all you are someone who has not taken the PU2 crash course, let me tell you, this crash course can help you. If you feel stranded, if at all you do not know where to begin, what to do and everything, and I told you the chapter's name, you can start doing these chapters with the help of the PU2 crash course, and it is just available for rupees 999. Here you will be getting chapter wise one-shot lecture. You will get the PYQ lectures as well. There will be subjects like PCMB plus Kannada as well as computer science if at all you need it. There will be lecture notes given to you, and there will be live doubt solving sessions as well. So if at all you are planning, make sure you go ahead and take it very quick, because the classes have officially started off. Okay, so I believe it will help you. And my dear children, let me go ahead with the topics that you should not miss. So from sexual reproduction, flowering plants, from the past 3-year analysis, these are the topics which are being repeated the most. So I have given like it 1 marker question has come from this. Along with that, I have to mention the structure of monocot embryo is also super important. Structure of monocot embryo, you might be asked to draw it. It's a super important question. So structure of pollen grain, as in you might be asked to make it, you might be asked to name the cells, you might be asked to tell the shape of the nucleus of each cell, you might be asked how many cells are there within a pollen grain, all of it. Sporopollen, all of it is super important. Apomixis, polyembryony, both of them are important. Examples of polyembryony is what? Citrus fruits and things like that. Apomixis, grasses and asteraceae, know the examples, know what is the process. Okay, now that is a topic that is being repeated. So one which I have highlighted in yellow, that means they have been repeated more than once as well. Now, features of flower pollinated by wind and examples, not just by wind, insect pollinated flowers, what are their features? They are also being asked. Please know the examples for all of this. For wind, it is grasses and corn-cob. Some people do not, remember it. So please go ahead and read through it. And as I said, structure of dicot and monocot embryo is super important, especially monocot. Artificial hybridization, you can be asked, what is that program that is taken by the farmers in crop breeding thing, which produces hybrid variety of plants. It is artificial hybridization, so you should know that. Okay, it is given in the NCERT, but we sometimes do not read all the details, right? So that is why we do not know. We think it's from some other chapter, biotechnology, principles and processes and things like that. Now, structure of anther, important to draw. Pollination and its type, autogamy, gytogamy, xenogamy. In that, in autogamy, you have clistogamous and chasmogamous flowers. Know the examples, oxalis, and all those things. Okay, viola, oxalis, fine. And false fruit, true fruit, and parthenocarpy, examples of it. Parthenocarpy shown by what do you call? Banana. False fruit example is apple. Why it is example of false fruit? All of this thing, super important. Double fertilization, post-fertilization events, as in what happens to the embryo, what type of seeds are being produced, or what happens to the endosperm. Does endosperm form first or embryo form first? So it is endosperm that form first. All of it, albuminous, non-albuminous seeds and things like that, and then the pollen pistil interaction. So these are the topics which are being repeated, which have come, which is why I have put it down. But that does not mean you should just go study these topics. Study the whole chapter, give more weightage to these topics. That is what I am trying to say. Okay, I am not here to make you lazy. I am here to guide you. Okay, guide you to prep smart. So, human reproduction, these are the topics which have been repeated most of the times, as you can see over here. Spermatogenesis, menstrual cycle, hormones in gestation and parturition. What are the hormones found in parturition and things like that? Male accessory gland. The questions have come. Mammary gland structure and diagram, super important. Events in gestation, month by month, what happens to the baby. In first month, this fifth month, what usually fifth month is being asked, to check that. Female reproductive system, again the diagram can be asked. Then the structure of the human sperm can also be asked. So these are the topics which are super important. Please make sure you specify them. Going ahead, we have reproductive health and principles of inheritance. In reproductive health, reasons for population explosion, mortality rate, and all these things. And apart from that, contraceptive methods, my dear children. In contraceptive methods, IUD are super important. It's most widely accepted method apart from oral pills, and also it is there for spacing children. So you should know their functions as well. Assisted reproductive technology as a whole, ZIFT, GIFT, AI, everything is super important. In ZIFT, how many blastomeres embryo can be put? It is 8 blastomeres, not beyond that. So all those things super important. Amniocentesis, this is given as a small line, but questions come from here also. Which are the diseases that you cannot find through amniocentesis? Jaundice is not something that you can find. Only genetic disorders can be found. Now, medical termination of pregnancy, when was the act being put and what is it about? When a person can undergo MTP and all of this. When is it safe? At 12 weeks is it safe, or basically 24 weeks, all those things. Now, sexually transmitted diseases, what are the ways you can prevent it? That is important. What is a sexually transmitted disease which cannot be cured? So 3 of them, hepatitis B, HIV, AIDS and herpes. Okay, and infertility, all of them start with H, which is why even I remember. So infertility and its reasons, a question which is usually not being considered, but it is being asked. So please look out for all these things. Principles of inheritance and variation, my dear children, you have lot of things, especially sex determination in honeybees. Incomplete dominance in snapdragon, as you can see, it has been repeated more of times. Chromosomal disorders, karyotype and symptoms, as in Down syndrome, what is a karyotype? Klinefelter syndrome, what is a karyotype? And Turner syndrome, what is a karyotype? What are the symptoms? All of it. Apart from that, pedigree analysis. One question usually comes, go through the pedigree analysis, and if you know the basics, you will be easily able to solve the questions. Linkage and recombination, this much percentage is given. Are they loosely linked or tightly linked? All those type of questions. Who put down gene mapping? Okay, it was Alfred Sturtevant who put down gene mapping. So small things comes under linkage and recombination. So don't just go for the term linkage and recombination. So many things are there within it also. Go through them. Pleiotropy and its example, phenylketonuria, super important. Co-dominance, A, B, O blood group, sex determination in human beings also, and also in insects. And Mendelian disorder, criteria of a genetic material, and then chromosomal theory of inheritance. All these topics are also important. And yes, As I said, there are other topics also important, but make sure you go through them really well. Fine. And apart from that, I think that's all. So Mendelian disorder, as in you have so many disorders, all of them being clubbed up. Color blindness is something that is usually being asked, and also the autosomal blood disorders, sickle cell anemia and thalassemia, all of it. So go through all of it. Now you have molecular basis of inheritance, these three topics being repeated, salient features of human genome, transcription and transcription unit, you might be asked to make it, discovery of the genetic material, how was it discovered, Griffith's experiment, Avery, MacLeod, McCarthy and Hershey and Chase, all of it, structure of a histone, what is there in a histone, arginine and lysine is there in a histone, central dogma, DNA to RNA, RNA to protein, all of it, features of a genetic code, super important. Now DNA fingerprinting steps and application, and the point mutation and frame shift mutation. Example of it, point mutation is what? Sickle cell anemia. So these type of questions have come, and as I've written over here, I have written the marks also for which it has come. So please go through them. Now chapter 6, evolution, Hardy-Weinberg principle and the factors affecting it, mostly has come. Evolution of man, super important. Evolution by anthropogenic action, as in the human activities, and your industrial melanism is an example for that. The moth experiment, no, that is also an example for this. What is saltation? Industrial melanism, I already explained. Homologous and analogous organs, example, they might be asking, this is happening, but what is this? Is it convergent, divergent evolution? All of it. Okay, and the Miller's experiment, here you might be asked to draw the diagram. Please try to draw the diagram. Super important. Okay, this diagram is super important. If at all you go for so many years back, again you might see that this question is being asked. Even though in the past 3 years only once, but still I am guaranteeing you it might come. Okay, so these are the things, and there is a picture for natural selection as well. For natural disruptive selection and all those things, please go through that picture also. Okay, and adaptive radiation, somehow it is not there, but I'm telling you, please go through these things. Adaptive radiation, the marsupials and the placental mammals, all of it. Okay. Then what do we have? Human health and diseases, my dear children. In that, all the diseases, their symptoms, their tests, super important. Plasmodium and its mode of action usually being asked, as in malaria and these things. Drug sources, mode of action and uses, all of it, super important. Cancer is the most important disease. Carcinogens can be asked, detection can be asked, what are the features of cancer cells can be asked, innate and acquired immunity, and in that come active and passive immunity, please go through them also. Lymphoid tissue, what are different type of lymphoid tissue, when does it become effector cell, where does it become antigen sensitive, all of it. Allergy and the mast cell, super important. Schematic representation of HIV action, this is a diagram, you should know this diagram properly, you might be asked to make it or might be asked to do fill in the blanks. Now structure of an antibody molecule, this is also an important diagram. Okay, please go through all of it. So these are the things that are usually being asked. And drugs I have written, right? And there is a thing that is, what is the effect of anabolic steroids? Anabolic steroids. So please go through the effect of anabolic steroids given at the end of this particular chapter. It is also super important. On females and males, it is different effect. Go through it. Now microbes in human welfare, enzymes made by genetic engineering. Okay, you should know them. And what else? You have biofertilizers, and in that the fungi, mycorrhiza one is super important, and nitrogen fixing bacteria is different from cyanobacteria, so know the examples differently. Root and shoot apical meristems, they are virus free. What are the things that you should be taking for producing virus free plants? It is the root and the shoot apical meristem. Then the biocontrol agents such as Bacillus thuringiensis, my dear children, and all the other examples, lepidopterans and baculoviruses, all of it are important. Go through it. As I said, in the Brahmastra series, I have explained them in detail such that none of your concepts are being missed. Even there will be concepts that are not over here. Okay, so please you can rely on that. Microbes in household products, your lactic acid bacteria and Saccharomyces cerevisiae, all of it. Microbes in sewage treatment. The sewage treatment totally is important. Please go through it. Microbes in industrial products such as beverages and other product, that is also important. So yes, so I have taken those chapters which I have put it over here. Now if you feel that you need the important topics of the other chapters as well, which I have not put, such as biotechnology principles and processes, biotechnology in application. Let me know in the comment section. I can help you with that as well. And over here, biodiversity and conservation, my dear children, in that in situ and ex situ conservation is one of the most important topic, and the causes of biodiversity loss. That is evil quartet, 4 reasons. So those 4 reasons are super important. Latitudinal gradient, pattern of diversity, where it is more, where it is less, all of it. And vertebrates with highest global biodiversity, like what do you call, fishes, which is more in number, and all of this thing comes, right? There is a graph being made, pie chart being made, go through it as well. Cool. So these are the very important things that I had to discuss with you. Make sure you complete these chapters. Whenever you complete, put a tick mark, go ahead with that. That is going to help you really well, and I can guarantee you that it gives you confidence. Okay, so that's all, my dear children. I hope it had helped you in some ways. And my dear children, please understand, as I said, do not think that I'll do tomorrow and never do it. Start from today itself, if at all possible, from now on itself. I hope you do it. Wishing you all the best. If at all you thought that this video was helpful for you, do not forget to hit the subscribe button right now. Ta-da, take care.